Hello, and welcome to the Chapter 16, Part 1 online lecture. You should use the information in this lecture to complete the Chapter 16, Part 1 guided notes, which, of course, you should complete before you come to class. Let's get started. This chapter is all about DNA, what DNA is, how it's put together, and what it does for the cell. Now, the structure of DNA and RNA should be a review for you. We went over that back in Chapter 5, but we'll go over it briefly in this chapter to make sure you remember all the stuff you need to remember. We're also going to discuss a little bit about how DNA actually stores genetic information, and then you're going to do a little bit of research to learn about how DNA was discovered. Let's review the structure of DNA and RNA. Both of these large polymers are in the nucleic acid group, and that means that they are both constructed of these little monomers known as nucleotides, which we'll take a closer look at in a moment. Now, DNA has kind of a classic shape. It's called a double helix, and that's because it's made of two strands of nucleotides that are wound together. We say it is double-stranded. The term DNA stands for deoxyribonucleic acid, and it, of course, is the molecule of heredity. This is the molecule that holds the genetic information that tells your cells what kinds of cells to be, how they should function, and makes you genetically unique among 6 billion human beings. RNA is considered the chemical cousin to DNA, and actually evidence indicates that it might be more like the grandfather or grandmother of DNA, that it might have appeared on the planet first. RNA stands for ribonucleic acid. By comparison, its shape is single-stranded, so it's just a single line of these nucleotides, and the bases on the RNA just stick out into space. They don't hook to anything. RNA plays a number of roles inside cells. Its major role is to help make proteins, and we'll see how that happens in Chapter 17. The nucleotide monomers of DNA and RNA consists of three major parts. We have the phosphate group. You can see the phosphorus in the middle there. We have a 5-carbon sugar, and we have this big molecule that's either going to consist of 1 or 2 rings, and we just generically call that thing a base or a nitrogenous base because you'll notice that it does contain quite a bit of nitrogen. So phosphate, sugar, base. I like to think of the sugar as being the heart of the nucleotide because it's really the molecule that connects everything together. Let's take a closer look at it. First of all, we have 5 carbons that make it up. There's carbon 1, carbon 2, carbon 3, carbon 4, and you might think that carbon 5 is over here, but that's actually an oxygen atom. Carbon 5 is over here. It kind of sticks out on a branch by itself between carbon 4 and the phosphate group. Now each carbon connects to something important. Carbon 1 is going to connect to the base. Carbon number 2 is either going to connect to a hydrogen or a hydroxyl group, and that determines what kind of a sugar this is. We'll look at that again in a moment. Carbon 3 is important because if this nucleotide connects to another nucleotide, it's carbon 3 that's going to reach down and grab the phosphate group of the nucleotide below. It might look something like this. There's our phosphate group, here's our sugar, if I can draw it, and here's our base. And it's carbon number 3 that will make a covalent bond between that carbon and that phosphate group. Carbon number 4 is found at this corner, and again, carbon number 5 is the one that will reach up and grab the phosphate group above it. Two kinds of sugars are found inside nucleotides, ribose and deoxyribose. Let's take a look at ribose first. This is the sugar that's found in the nucleotides of RNA. Now I told you that carbon number 2 determines which kind of a sugar this is. So let's see what's attached to carbon 2. You'll notice that in ribose, an OH, a hydroxyl group, is attached to carbon number 2. That's what makes it ribose. In comparison, deoxyribose, which is found in the nucleotides of DNA, only has a little hydrogen attached there. So it's lacking this oxygen that ribose has at this position, so we call it deoxyribose. It's like ribose, but missing an oxygen. When we deal with three-dimensional objects in science, it's not uncommon to give the different sides of that object different names. For instance, when you deal with the human body, you learn that the chest and the belly side of the body is actually called the ventral side of the body. And the back, where our shoulder blades are, that's called the dorsal side of the body. Why do we do that? Why do we give everything special names? Well, it's not just to use a lot of fun words and give things new names. It's actually to help make our language more precise and to avoid confusion. When you're dealing with a human patient, terms like front and back can actually be a little bit confusing. Now, nucleotides are the same thing. These are three-dimensional molecules, so we can't say top, bottom, right, left side. We need to give the sides of this thing particular names. I like to think of the sugar of the nucleotide as looking like a little house you might have drawn when you were a kid. The foundation of the house, the base of the house down here, is called the 3' end. Now, why is that? Well, it's because the 3 carbon is on that side. It's at the base of the house. The roof of the house up here is actually referred to as the 5' side, and that's because it's closest to carbon number 5, which is over here. When we use these nucleotides and build them into long strands, those strands are also going to have 3' and 5' ends. Here we have the phosphate group. This diagram actually shows a simplified version of the phosphate group, and that's because when these nucleotides are floating around by themselves, they typically have 2 or 3 phosphate groups attached. When the nucleotides join up and make a polymer like DNA or RNA, they tend to lose those 2 phosphate groups. So we're just going to show them as having just 1 phosphate group. The phosphate group attaches to the sugar by carbon number 5, which is shown there. In addition to containing different sugars, nucleotides can contain different bases. There are 5 different bases and they fall into 2 categories, the purines and the pyrimidines. All of these structures have rings. Purines are double-ringed structures, so you can see there's 2 rings hooked together. The purines include adenine, represented with a letter A, and guanine, represented with a letter G. The pyrimidines are single-ringed structures, so an easy way to remember that one of these is a pyrimidine and falls in that category is that its shape is round like a pie. An easy way to remember which bases fall in this category is to remember that pyrimidine has the letter Y in it, and so does cytosine, which is represented with a C, thymine, which is represented with the letter T, and uracil kind of messes up this memory trick, but if you imagine a U as having a tail, it makes a Y, so it's a pyrimidine as well. Different polynucleotides favor different bases. So in DNA, you'll find nucleotides with adenine attached, thymine attached, cytosine attached, and guanine attached. Whereas in RNA, you'll find nucleotides with adenine, uracil instead of thymine, cytosine, and guanine. The nucleotides are named for the bases they contain. So a nucleotide that contains adenine will just be called adenine. Here we have 3 different views of a DNA molecule. DNA is a polynucleotide, which means that it's constructed out of nucleotide monomers, 2 long strings or strands of nucleotides hooked together with bonds. If we take this diagram of DNA and flatten it out, you can see the nucleotides and how they interact. Here, for instance, is one nucleotide with the sugar phosphate and base, and you can see that it's connected to the nucleotide below it, and so on, to make these long strings. Notice that it is the number 3 carbon right here that reaches down and attaches to the phosphate group of the nucleotide below. If we remove the bases from a diagram of DNA, it's easier to see the 2 backbones. Here we have the backbone of one strand, and here we have the backbone of the other strand. Now this backbone consists of the sugars, and this D stands for deoxyribose in this case since it's DNA, and phosphates. So sometimes collectively this structure is referred to as a sugar phosphate backbone. Now notice a couple of things about this backbone. One, it's held together strictly with covalent bonds. Notice all these solid lines holding it together. And covalent bonds are nice strong bonds. So this is a really strong backbone. The other thing to notice is that this backbone repeats over and over: phosphate, sugar, phosphate, sugar, phosphate, sugar. So what that tells you is that the backbones do not store any genetic information. The backbones are just there to give the DNA structure and hold it together. But if this were a book and you were reading it, the words in the book would say sugar phosphate, sugar phosphate, sugar phosphate. And that doesn't give us any information. Next, let's examine the bases that hold these two strands together. Notice that the two strands are not identical, nor are they mirror images of one another, but they do fit together in a particular repeating pattern. We call this pattern complementary base pairing. The rule goes like this: adenines will only bind with thymines. So anywhere you find an A on one strand, across from it you're going to find a T. Also, guanines will only bind with cytosines. So we say the rule is A with T, G with C, also known as Chargaff's rule, named for the scientist that characterized this. Now notice what kind of bond is holding these bases together. We have dotted lines here. That represents hydrogen bonds. Hydrogen bonds, if you remember, are weak bonds. So we have strong bonds holding the backbones together, but weak bonds holding the center together. Now that's important. Because there are weak bonds in the middle here, that means that the DNA strand can actually open. When the cell needs to use those genes, the DNA strand will open. When the genes are no longer being used, they can close up and those bonds can hold them together and keep them safe. So why does A only pair with T and C only pair with G? Well, remember that these molecules are three-dimensional. They have specific shapes. They also have specific functional groups that stick out. And because of that, thymine can only bind with adenine. Notice that thymine and adenine make two hydrogen bonds with one another. Also, guanine can only bind with cytosine. And notice that G and C make three hydrogen bonds with one another. Another important thing to notice is that thymine as a pyrimidine is bonding with a purine, adenine. And over here, that pattern continues. Guanine is a purine. It's bound to a pyrimidine on the other side. Now, why is that? Why would you want to have one of one type on one side and one of another type on the other side? Well, because one has two rings and one has a single ring. If you put them together, it means that DNA has this kind of uniform structure to it. If we tried to put two purines together, because they have double rings, it would make lumps in the DNA, and the DNA is not going to function as well if it's all lumpy like that. RNA molecules are similar in structure to DNA molecules in that they are long strings of nucleotides. But RNA is single-stranded, whereas DNA is double-stranded, and RNA is going to use uracil instead of thymine. Now here's an interesting thing that RNA can do that DNA cannot. Because RNA is single-stranded, it can fold on itself, and so it can begin to form these interesting three-dimensional structures. So RNAs fold up sort of in the same way that proteins fold up, and so RNAs can have a number of different jobs inside cells. What's holding their structure together? Well, it's complementary base pairing. Because RNA has uracil instead of thymine, adenines are going to match up with the uracils, and cytosines and guanines are going to match up as we saw in DNA. So if we have sections such as these that are complementary to one another, they can bond with one another, and those hydrogen bonds help hold the RNA in its shape. Here we have a type of RNA called tRNA. It very conveniently has a kind of T shape to it, but it's going to play a role in helping us make proteins, and we'll see what it does specifically in Chapter 17. Something you may have noticed if you looked closely at some of the previous DNA diagrams is that the two strands of DNA do not run in the same direction. DNA is put together a little bit like a two-lane highway. So if you imagine a highway as being sort of like Mopac or I-35, when traffic is moving, the traffic on the two sides of the highway are going to move in opposite directions. DNA is like that too. Notice that if we look at the nucleotides on this side of the strand of DNA, that the little houses, the little sugars, are sitting on their foundations. On the other side, the other strand, those sugars are flipped upside down. Now, this is science class. We can't say they're just flipped upside down. We need a sciencey word for it. So we say that the two strands of DNA are anti-parallel to one another. Because the nucleotides are three-dimensional and have sides, when we put the nucleotides together into long strands, the two strands are going to have different sides with the same labels. This nucleotide is sitting on its foundation. We call that the 3' side. That means that this strand has a 3' end. This is the 3' end. On the other end, we're on the little roof side of the house, and we call that the 5' side. So this strand has a 3' end and a 5' end. Because the other strand is upside down or anti-parallel, the 3' of this strand is going to be on this side, and the 5' is going to be down here. Now that we've refreshed your memory on the structure of DNA, we can now begin to discuss how DNA actually stores information. I mean, think about it. These molecules inside your cells are supposed to hold enough information to tell each cell what kind of cell to be, to control its activities, and to control the activities within your body over the entire lifespan of your life. Unless you have an identical twin, your DNA also is unique. It makes you genetically unique among all other life forms. No other form of life has your exact sequence of DNA. So where is this remarkable information stored? Well, let's look at a section of DNA. A section of DNA such as this might be called a gene. And a gene is really a recipe for making either an RNA molecule and or a protein. So we can make RNAs or proteins based on this information. Where's the actual information? Well, it's here. It's in the bases. Remember, it's not going to be in the backbones because the backbones just repeat over and over. But the bases have a sequence, and it's the sequence of those bases that's important. What the cell will do is it will open up DNA, it will choose just one of the strands, and different genes have different strands with information, and it will read this strand, for instance, from top to bottom. It'll read the sequence adenine, cytosine, guanine, thymine, and so on. And because the sequence of those nucleotides differs from one gene to another, that can store information. Think about reading a recipe in a book. The order of the words used in that recipe is really important. If you change the order of the words around, it can change the meaning of the recipe. Same thing applies here. It's the sequence of the bases that stores the information. At this point, if you're thinking that it's pretty implausible that a molecule as simple as DNA could store all of that information, you're definitely not alone. Back in the 1950s, when DNA was discovered, scientists were shocked at how simple it was. I mean, it's only made of 4 different types of nucleotides in these repeating patterns. So scientists thought, well, there's no way that DNA is complicated enough to store genetic information. So for a long time, they thought that something more complex, things like proteins, which are really complicated, must be where our heredity lay. And yet, it turns out that DNA really does store that amount of information. To explain how this works, I want you to think of DNA as being a molecular language, a chemical language, and it's a language that only has 4 letters. Now, 4 letters isn't very many letters, especially compared to a language like English. English has 26 letters, and think about all the things we can express in words using our combinations of 26 letters. Well, English is a pretty complicated language, and there are simpler languages. Consider this: Hawaiian only has 12 letters in its alphabet, and you can express anything in Hawaiian that you can express in English. There are even simpler languages. Binary language, the language of computers, really only uses two symbols, zeros and ones. So there's an even simpler language that only uses two characters to express things, and you can express everything in binary that you can in English. So a language such as DNA actually falls kind of in the middle. With only four letters in the alphabet, you can express everything you can express in English, essentially. And you can express all of the information you need to run a cell and to run an organism. Incidentally, I still occasionally hear this argument that DNA is too simple to store the genetic information of life being tossed around. Not among scientists, mind you, but by groups that, let's say, just wish evolutionary theory could be disproved. The idea is that DNA is too simple to store all of the genetic information that makes everything from giraffes to fungus to bacteria to people, so that some sort of a creator or an intelligent designer must have been involved in the formation of life on this planet. Now that you know a little bit more about DNA, I hope you can see the logical flaws in this argument. Even though the language of DNA only consists of four letters, you can still store a lot of genetic information using those four letters. The secret to this system is really in the sequence of those letters, not in the number of the letters. Let me show you an example. I'm going to draw out a sequence of nucleotides that is 10 nucleotides long. These are A's. They may not all look like it. There we go, 10 A's in a row, 10 adenines in a row. And let's say that I was just going to try and see how many of these sequences I could come up with that are 10 letters long in length and are only use those four nucleotide letters. Well, here, this first one is all adenines. Maybe the second one is 9 adenines and a thymine. Maybe the third one I come up with is 8 adenines, a guanine, and a thymine, and so on. If I kept doing that and was really, really persistent, I could come up with over 1,000,000 sequences that fit those rules, that are only 10 nucleotides in length. Now consider this. The sequences that code for most proteins and RNAs aren't 10 nucleotides in length. They're hundreds of nucleotides in length, or even thousands of nucleotides in length. When you have a sequence of that kind of size and you're looking at the sequence of the letters involved, you can code for a whole lot of different kinds of proteins and a whole lot of different kinds of RNAs. You can literally code for millions of different things because the sequences differ. We're going to look at how the cell actually extracts information from these sequences more in Chapter 17, but I'll give you a little introduction for now. In our chemical language, I want you to think of the individual letters as the nucleotides. So the A's, T's, C's, and G's, or A's, U's, C's, and G's, those are our letters of our language. If you put 3 of these letters together, you get a word in this language, something called a codon. So what the cell will do is it will read these sequences, and it reads them in groups of 3. And each group of 3 means something. Each group of 3 codes for a specific amino acid. And if you put the right amino acids together in the right order, you can make a specific kind of protein. This entire sequence with all of its letters and codons might be one story or one recipe in a book. So that little sequence is called a gene. Again, a gene is a recipe for making a specific protein or sometimes RNA. One thing that I hope you take away from this lecture is really just how simple the structure of DNA is. It's really just 2 strands of nucleotides that fit together in a very predictable pattern. However, even though the structure of DNA seems simple by today's standards, the discovery of DNA, that story, is far from simple. The discovery of the structure of DNA was really the Human Genome Project of the 1940s and 1950s. And by that I mean that there were a number of labs and many scientists trying to figure it out, and they were all competing with one another. Now, each different lab was able to figure out a little piece of the puzzle, but no one could really put together the whole story and figure out the whole structure until these 2 guys named Watson and Crick came along in 1953, and they put together all the information and figured out the overall structure of DNA. Now, this story is very controversial because Watson and Crick essentially stole a lot of data from other scientists. Some of it was shared with them legitimately, and some of it they just stole. They were known for doing things like slipping into the back of research presentations and making a few notes and slipping out before anyone knew they were there. I really don't do this story justice, so what I'm going to have you do is do some reading in your textbook in Concept 16.1, and I'm also going to have you visit a website called Vision Learning and read about these different scientists, read about their different contributions, and take some notes on that. So that is included in your guided notes, and I do want you to complete it before you come to class next time. We're going to work with it some more. All right, you guys, you're finished with Chapter 16, Part 1. Make sure you complete your notes, try all your turns yourself before you come to class, and then bring that stuff to class so we can dig into it and go over it. Good job.